say polymer and polysaccharides or monomers and monosaccharides? What do you think is the difference? Think of the word polysaccharide. The word saccharide means sugar.
Cutie bear.
Yeah, exactly. So when you say polymer, you're not saying which kind of polymer it is. You're not saying if you're talking about a sugar or you're talking about a protein or you're talking about a nucleic acid. So right now, for you, this is the only polymer you know, so you're thinking it's the same. But when you know other polymers, like carbohydrates are a polymer, but you have proteins that are polymer, nucleic acids that are polymer, there's many materials that are polymers. So then now those have specific names. So polysaccharide is a polymer of a sugar. It's a necklace of polymers, yeah. A necklace, yeah. It's a, oh, you mean a branch of polymers? Well, a specific example of a polymer. Exactly. Okay, guys, any more questions? Please write them down before we move on to now the more long examples of polysaccharides. I put some videos in there. We're going to address questions, have a discussion, learn about polysaccharides. That will take us quite a bit of time. Then I give you a practice activity. We'll do it and show you how to do that. Then after that, we'll discuss a little bit about inquiry, because tomorrow we're doing an inquiry, whole period for inquiry. So you're going to practice inquiry, you're going to finish your lab design. We're going to do a short quiz for inquiry, so that make sure that's clear in your mind how that works. And then hopefully if you finish your lab on Friday, you'll run it after our quiz. So yeah, it'll be exciting. Okay, so that's what we're doing today. But for now, before we get going into more complex ideas about carbohydrates, I would love to hear questions. So if you went through your notes yesterday, if you had a chance to look at the PowerPoint from what we learned, there must be parts that you're still a bit unclear about. And even though I will recap, I would like to hear the parts that you are confused or unclear about. There were a lot of words you had to learn, right? So take a minute to go through your notes, to write down the questions here in the chat before we get going, okay? The class is almost fully present here. Almost everybody has shown up already, but please do write questions that you may have about stuff you don't understand in the chat, okay? I'll give you a few seconds. I'm just going to check out what I was checking emails, see if somebody has called, said they were not going to be present or anything like that.
Wait, miss, I have a quick question.
Absolutely, yeah.
Yeah, I think you should probably go on your Google, like the PowerPoint though, because so I think one of their PowerPoint on one of the slides, I don't know how to pronounce this word, but it's called like glycohydrate, like
Glycogen?
Yeah, I think so. Yeah, and it said it's an isomer of hydroacetone. Which one is like?
Oh, glyceraldehyde.
Pardon?
Is it glyceraldehyde triphosphate? Glyceraldehyde is an isomer of dihydroxyacetone.
Yeah, that's how you say it, glyhydrate, right? It's an isomer.
Glyceraldehyde.
Which one was the hydroacetone again?
Dihydroxyacetone. So glyceraldehyde is an aldose. Dihydroxyacetone is a ketose.
Oh, so it's an
and they're both trioses. So 3 carbon sugars.
Oh, okay. So wait, so you know how I said, wait, how do you say that word again? I can't like.
I know it's very tricky. Glyceraldehyde. So glyc for sugar, aldehyde, glyceraldehyde.
Oh, yeah. So you know how I said it's an isomer of hydroacetone? It's actually dihydroacetone, right? Not just not. It's not just hydroacetone, but it's dihydroacetone, right? Okay.
Yeah. Uh huh. What do you want to know about them being isomers of each other?
Yeah, so which isomer is it again? Is it the one with like what's it?
Oh, so anything that has the same formula, so anything that is C3H6O3 is isomers of each other. Anything that is C5H10O5 is isomers of each other. And anything that is C6H12O6 is isomers of each other because they
Oh,
have the same formula. So anything under the triosis is going to be isomers. Anything under the pentosis is going to be isomers.
oh, so like pentosis has ribose and then ribulose, and like those two are isomers.
Exactly. And anything that they give you the same formula, you know it's an isomer because anything that has the same formula, no matter how you rearrange it, we consider an structural isomer.
Would it be like which isomer would it be like the out of three, like structural and like which one would out of
Structural.
for
It's
all of
all
them?
structural.
Okay, okay, makes sense. Okay.
Yeah, because they're not 3D mirror images, they're not, and you don't have a double bond cis and trans. So these are all structural. They simply, you've moved one thing or something.
Oh, so basically anything under triosis, pentosis, hexosis. Oh my god, saying these words are hard, but like anything under them, they're basically structural isomers.
Of each other, yeah.
Yeah.
So
Okay.
all the six, all structural of each other. All the three, all structural, yeah.
Okay. Yeah, that's it.
Thank you for asking that question, Sebastian. I'm sure other people don't understand it and they're just feeling a bit shy about asking.
All right, no
So,
problem. I just wanted to make sure.
and anybody else, guys, please do write your questions or say your questions because it's helpful when you take a second to say the stuff that you're not clear about. It helps you think about it. It helps you understand. I am sure people not asking questions doesn't mean that everybody gets it perfectly. I am pretty sure. I think it's because people either are feeling shy or they haven't had a chance to review, so everything is feeling like an ocean of information, which is a dangerous state to be in because this thing moves very quickly, right? Like this week we're covering 2 macromolecules.
We miss
Next
out.
week we're covering probably another 2, and then we're done. It's exam. It's test.
I also
Sebastian, yeah?
I have another quick question. What's the difference between the aldo how do you say these words again?
Aldose and
Yeah, aldose
ketose.
and ketose. What are the difference between those two again?
Okay, so because that's a bit simpler, I'm going to leave somebody in the class to say that. What is the difference between aldose and ketose? Who thinks they can answer that question? What is different between those two types of sugars, aldose, ketose? Good job, Hiva. Exactly, Rihanna, Ethan, nicely done. Thank you. So exactly. So all sugars have a carbonyl functional group. But if the carbonyl functional group is at the end, we say that's an aldose because molecules that normally have a carbonyl group at the end, we call them aldehydes. So the name comes from that. And then molecules that have a carbonyl group somewhere in the middle, we call them ketoses. So that's how come it's called ketones. So that's how come the sugar version of that thing is called ketose.
Oh, okay.
That's
So the carbonyl
that's
group.
where they come from.
So like I said, the carbonyl group is at the end. Then basically it's aldosis. But then if it's basically in the middle, then it's ketosis.
Ketose.
Okay.
Yeah.
Yeah, that makes sense.
Okay, so lovely. So then let me see if there's other questions. Functional carbon groups in the middle. Yeah, very nice. People came to the rescue. I'm very impressed. That's awesome. Okay, who else has another question from yesterday before I start and review the content that we were doing? Can you over the ring and linear? Yeah, I'll review everything that we did yesterday for sure. I always go over the basic stuff that we just did. I just wanted to know if there was anything else. Okay, if there's nothing else, okay, I'm going to go back to the PowerPoint that we did yesterday. Can you explain Sunny's question? Sunny's question. It's a ring, where would be at the, if it's a ring, where would the end be? Oh, well, when I go back to the ring, I'll show you. That's a very good question. But then a ring derives from ketose or an aldose. So if you have a ring made from an aldose, it looks different than a ring made from a ketose. Okay? But the question is, what would be the end? There's no end. Okay, the end, when we talk about aldose, that comes from the linear form of the molecule. It doesn't make sense to talk about that in the ring form of the molecule.
Wait, miss, sorry, I have another really quick question. It's called aldoses. You know the end, right? How do you know where, like, which one's the end again? Like, how do you know where is the end? Like, is it the bottom or the top? Like, where do you know?
Oh, there's no bottom or top or end, because these things are all floating in the. But what I mean is the carbonyl group can be somewhere in between the atoms, or is at the beginning of the atom. So if the chain has a carbonyl group at one of the two endings.
Oh, okay. So it doesn't matter. It's just like if it's in one of those ends.
There's no up or down or left or right.
Oh, okay, got you.
No.
Okay, I see.
Good question, though. That's a good question.
Thank you.
Okay, so Sonny, does that make it clear? Like, there's no beginning or end. When it's a cycle, it's just, the cycle is a consequence of the original linear molecule, and the original linear molecule did have aldose or ketose. Does that help Sonny and Olivia? Hopefully. Khadija saying no. Khadija. Yeah, I know, Khadija, you want me to show you the full ring. I know, I have to show you with a PowerPoint. Yeah, okay, so let's go back and I'll show you what I mean. So let's do presentation. And then I'll go back and we'll review everything. Okay, so when we go to our classroom, by the way, I don't know if you've noticed, just to bring to your attention, number one, I posted here lessons that are every chapter is in, not lessons, for every chapter, there's information here from the textbook instead of like before, the way I shared it, you couldn't open it. Also, there is a cool video here about cotton that about cotton that is really worth looking at because it helps you with what we'll learn today. This, what you see here, is a recording of the lesson that we are going to learn today, which I recorded in the morning in case somebody was away. And then for classwork, this is our project and this is a handout we're about to do. Okay, so I'm going to go to our PowerPoint. So if you don't know where it is, it's here. Okay, this is our macromolecules PowerPoint that we started yesterday. And we're simply going to go from where we started quickly. I'll go through an review and pause in the part of the rings. Okay, so what we started learning is about macromolecules, which is the rest of the unit. And in order to understand macromolecules, first we paused and learned about the concept of polymers, because 3 of our 4 macromolecules are made of polymers. So we said that a polymer is a long chain of molecules joined together. And we said there's 3 kinds of macromolecules that are made of polymers: carbohydrates, proteins, and nucleic acids. I showed you how a polymer is made, and the reaction that makes it is called a dehydration. And it simply consists on taking the hydrogen and taking the hydroxyl from both ends of the molecules and joining them together to make a longer polymer. And this is an hydrolysis, which is a reaction that breaks down the polymers. Then we went on to learn about carbohydrates, which are the macromolecules that include sugars. And we learned details about them. We didn't go to polysaccharides, but we learned about monosaccharides. We said they all have the formula CH2O, which is what has the reason the carbohydrates are called carbohydrates, carbon, water. And then we chose to talk, for instance, about glucose, which follows that pattern, C6H12O6, as very common monosaccharide. And then we learned about 2 kinds of monosaccharides: the aldoses, which have the carbonyl group at one of the extremes, okay? And the ketoses, which have the carbonyl group somewhere in the middle of the molecule. And within the aldoses, we looked at a few examples. So we had some examples of trioses, like glyceraldehyde. That's the one that Sebastian was talking about. This is a triose, 3 carbons. Then we had the pentose ribose, 5 carbons. And for hexoses, we looked at good old glucose and its isomer galactose. And so what Sebastian was asking was, are all these, the ketose for 6 carbons and the aldose for 6 carbon, are all these guys isomers? Yes. Anything that has this formula is a structural isomer of each other. So any hexose is a structural isomer of each other. Any pentose is a structural isomer of each other. And any triose is a structural isomer of each other. And this is the other molecule that Sebastian was mentioning, dihydroxyacetone. And it's simply just another triose. And we have ribulose and fructose. Okay? And then we talked about how these molecules make a ring. And that's what it wasn't Hiva. Who was asking this question? Wait. Khadija was asking. Khadija was asking, how is it that the ring form works? And so let me pause and explain. So for instance, in this example that we're looking at, this is glucose and aldose. This is the carbonyl group. Carbonyl group, because this is a functional group, is the carbon number 1 and the rest are numbered sequentially, okay? And this glucose, when it's folded, will look like this. You'll have the first, second, third, fourth, 5, and 6 carbon. But what occurs is this OH in the 5th carbon becomes quite interested in this. You can say there's an attraction. And this generates a corner that cycles the glucose. Now, in monosaccharides, this cycle form and linear form will in equilibrium. They will exist a little bit of time in the cycle form and a little bit of time on the linear form. But later on, when you are bonding two different monosaccharides, now the only way they bond is in the cyclical form. So to go back to, I think it was Sunny that was asking, well, where is the beginning or the end in a cycle? You don't have the beginning or the end in a cycle. What you have is simply carbon number 1 that used to be the carbonyl group, now is this. Okay? Now, that's the ring structure that you have for a hexose. But if you had a ketose like fructose, the ring structure looks a bit different. Hopefully that clarifies things. I would like to hear if there's any more questions. What form do they bond in? That's what we're about to show. Okay? So once you have learned how the ring structure happens, then you are going to learn how they bond. Okay? How we go from a monosaccharide to a disaccharide or a polysaccharide. So the bond that is formed by dehydration, we learned earlier about how a polymer is made. The bond that is made by dehydration has a specific name in sugars, okay? So over here, you learn about how dehydration makes bonds. These are general bonds, but in sugar, they have specific names. Just like when you say polymer, that's general, but polysaccharide is a polymer of sugars. Then the dehydration bond formed in sugars has a specific name called glycosidic linkage, okay? And that's only for sugar. Like I said to you, glyco means sugar. So that's the name of the bond in sugars. The glycosidic linkage bond forms when you have the ring form only, okay? You will never have a glycosidic linkage or a polymer or a disaccharide made with linear sugars. They are the ring that connects. And you always will have a bond. The glycosidic linkage can form between different carbons in the cyclical sugars. So in these two, glucose with glucose join together to make maltose. In this very example, the carbons that are connecting to make the glycosidic linkage are the first carbon of the sugar and the fourth carbon of this sugar, and that's why this is called the 1,4 glycosidic linkage. Okay? Because this is the first carbon and the fourth carbon. In this example, you have glucose and fructose, and this connects the first carbon of the glucose and the second carbon of the fructose. So this is a 1,2 glycosidic linkage. This is where we ended the presentation yesterday. So before I start new content, any more questions? So dehydration is a reaction, glycosidic linkage is a bond. The dehydration of sugars to join two monosaccharides makes a glycosidic linkage. Okay, Shani? So it's not exactly, the glycosidic linkage is the name of the bond. It is still a dehydration. Yeah. Okay, any more questions about the stuff from yesterday before we go into new content? All right, so to continue, what we left it here, we were going to start polysaccharides. Now, polysaccharides, as you can imagine, is a continuation because we just learned how two things bond. So obviously a polysaccharide is a long chain made up of many of these. So what occurs is that they are all going to be joined by the glycosidic linkage, but now you have many. So polysaccharides have many jobs, many functions, okay? And their functions are to do directly with how they are connected and the type of glycosidic linkage they have, okay? So for instance, the first more obvious function is the function of storage. So some polysaccharides act as storage molecules, okay? And this starch is a very typical storage molecule. So here you have two examples of storage polysaccharides. In plants, in the chloroplast, you may have starch nodules that in some plants may contain amylopectin, which is a storage polysaccharide, or amylose, which is cotton, the sugar that you find in cotton. And this is in the video that I put in the Google Classroom, by the way. And so this is a storage polysaccharide for plants. This is glycogen. This is a storage polysaccharide for animals, human beings included in here. Okay, it's an animal kind of polysaccharide. And you can see how it works because you can see the little rings that are all making this long chain. So you can see there are simply many long chains of this thing. Okay? So before I continue, I would love to ask you a few questions to make sure that you're with me and you understand. So he said, why do they have the curly fry shape? Okay, so curly fry shape. We'll talk about the curly fry shape in a little bit, okay? Because that really matters for the next discussion. But we park that for a sec, and let me ask you another question. So you saw that plants have storage polysaccharides like starches, amylopectin, amylose. They use them for storage. So my first question is, plants do photosynthesis. Why would plants need to store energy if they have like a factory of energy whenever they need to? Why would plants need this? For the night when they can't get sunlight as easily. So that's the very good, clever solution. I definitely appreciate that. Now, don't forget that the night is a short period of time, right? We don't eat at night either. So, but that's not a bad solution. What else would they need to store? So for what Sunny said, for when they cannot photosynthesize. So for instance, Justra said for the winter, that's a very good idea. Zoe as well, for the winter, exactly, there's no sunlight. So that's a good idea for the winter. Now, what I want you to think is that for a minute, if you think of all these starches, think of the food you eat. Where do you find starches? What foods have starches? So one thing is for the seeds, like fruit. So fruits have a lot of starches, right? And potatoes have a lot of starches. So let's think, why would fruits have starches? Why would potatoes have starches? So, so in the case of seeds, exactly, when the plant is just a seed, there's no leaves, right? The plant has no way of making energy. So it needs to grow into a stem and a leaf in order to develop the ability to make energy. And then what about potatoes? You said growing underground. What are potatoes? What part of the plant? Exactly, they are roots. So exactly. So the root obviously has no leaves, and the root has to survive maybe in the winter. So there's going to be storage for when there is more energy, or maybe as it's developing, it's going to be storage. So all these are the reasons that plants require storage. Now, in our case, if you looked at there, we said, okay, there's an energy storage solution for us, a polysaccharide that we use called glycogen. But glycogen is a short-term storage. What is our long-term storage for a long time? What do we do for a long period of time? If you don't have energy for a long, exactly, good job. Exactly, we use lipids, we use fats, right? Because long-term, we use fats. But we use polysaccharides for short-term storage. So the way to think about it is like this. Say that I went to a marathon, right? Like I'm running a marathon, okay? Like it's a long time. I don't know if you've ever seen a marathon, but we're talking, if you've ever gone for a run, I don't know, do any of you guys run? Have you run more than 2 kilometers? Olivia said, I don't like running. I mean, not many people love it. Running is only fun if it's not freezing and if you have really nice music. But I really admire people that can run for a very long time. I used to, Kevin bikes. Biking is also fun with music. It's nice. Sometimes I join the track and feel grade 5, 6, and never again. Well, I know many kids that join when they're younger, but then you get so busy too. It's tricky, right? I used to run when I was younger, but my knees cannot take it anymore, so I just work out for short periods of jumping, or I cannot run for a long time. But I used to run, but never. Holy cow, those 35-kilometer marathons! Whoa, that is really impressive. Anyways, so say that you were running a very long marathon of 30 kilometers, right? What do you think will be the first energy you use? What kind of food? The second energy you use when you're like, okay, I'm running out of energy. The third energy you use when you're like, okay, it's desperate. And the last when you're like, okay, this is really bad. So think about that. Like the first energy when you're like, okay, you're good. So first, Olivia said carbohydrates, but which kind? Which kind of carbohydrate will you use first? Oh, Nicole summarized it. Sugar, fat, then protein. Okay, so let's see. Within sugar, which do you think you use first? Do you use just polysaccharides? What else do you use? So exactly, you use glucose. You have glucose in your blood. So when you, like say you had orange juice or whatever, that's the first thing you consume. You consume the glucose in your blood. Then I've run out of that. Like I'm now hungry, so I'm going to use the glycogen, which is normally stored in the liver. Then I've run out of that. Then I'm going to use lipids, the long-term stores that I have in my body. And then after that, your body starts kind of in desperate circumstance consuming itself and its own muscles, which is not healthy and is not okay. And it only happens if you're really undernourished and in bad circumstances. Because what happens if you eat your own protein? What are you eating? What part of your body are you consuming? Where is the protein in your body? Oh, I didn't make the order. Olivia made the order. Sugar, then fat, then protein. But it would be glucose, glycogen, fats, and then protein. And Kevin clarified, protein is in your muscles. In your muscles. Nice job. Nice job. So your protein is in your muscles. Now, what's the problem with consuming your own muscles? What's an important muscle that you don't want to be consuming? brain. Brain is not muscle, Olivia. Don't you remember grade 10, the tissues? Um, Diaphragm, more important than that, man. Of course, diaphragm is important, Grace, but I'm talking about very important. Oh, Osime said this. Heart, heart. That's really bad, right? So when people are very undernourished or very sickly, or if they have maybe anorexia nervosa, what ends up occurring is that the malnutrition makes them consume their own muscles, and they consume their own heart muscle, and they end up dying of a heart attack. So yeah, yeah, it's kind of, when I told this to my period one class, they were like saying, "You're eating your own heart. Eat your heart out," kind of a comment. Yeah, both Sean is smaller and weaker, and yeah, that's kind of terrible. And that's why we have to eat healthy and exercise, but don't starve yourself. It's really bad. So the order. So what's the order? Glucose, glycogen, fats, and then protein in your muscles, which is not healthy and shouldn't happen. Can you poke a hole in by digestion? I don't think you would necessarily just poke a hole. It simply will get weaker, I think. But I'm not a cardiologist, so don't take me by my word. Which ones are the lipids again? Well, you really haven't learned much about the lipids because we haven't started that. We're starting that on Thursday. But lipids include fats. How small can a heart? That I don't know, Sean. I don't know, sweetheart. Okay, should we continue? Really fun discussion. Okay, a bit green, but this is good learning. It's biology. Okay, so we were saying that these are storage polysaccharides. Examples of these storage polysaccharides are glycogen, which is in our liver and in the liver of most animals. Amylopectin and amylose are starches that plants can store. And then we said a glycogen is short-term storage that occurs in animals in the liver, okay? And sometimes in a bit of muscle cells. So now there's another kind of polysaccharide which has a different job. It's a structural polysaccharide. So instead of being a polysaccharide that is simply about storing energy, this is a polysaccharide that is to do with making structures for plants, protection, not just for plants, for animals sometimes. So they have like protection. They tend to be much more rigid. Okay? So what I wanted to show you is that, for instance, cellulose is a very important polysaccharide. It's a polysaccharide that is important in terms of protection and is major component of the cell walls. You may have even heard that before. So cell walls, we know protection, structure. So what do you think will be the difference between these structural polysaccharides that are protecting the cell wall or protecting the cells, or in the case of animals, for instance, insects, they are this exoskeleton of insects. So this is a different job than storage. So what do you want to be different in these molecules? Storage of energy, where I want to access the energy, versus something that is protecting me. How would you think these two molecules should be different? So exactly. So for protection, you would suspect, Simavera said, more dense for protection. So, or stronger bonds, says Felicia. Exactly. You must have to have something that is more rigid or more strong, while if I want to access it, I want it to be easy to break down. So less solid, right? Very good thinking, very good prediction. And this is exactly what occurs. So important to take note, for instance, in the case of cellulose, this polysaccharide has two different kind of glucoses, okay? Two ring forms of the glucose. So the glucose can be alpha or beta, and I'll show you what the difference is. The one that you saw before, when you saw the glycosidic linkage 24, that was alpha glucose, okay? The same glucose can make two kinds of rings. this is an alpha glucose ring to the left, you can see that the hydroxyl group goes down. If you make a beta glucose, the hydroxyl group ends up going up. Well, in the first carbon now, you have one with a hydroxyl group going up and one with a hydroxyl group going down. What do you think will occur in the glycosidic linkage? How would it be different? So one of them has the hydroxyl going down, the other one hydroxyl going up. How do you think that will change the actual glycosidic linkage? Nobody's taking a risk. Nobody knows. Oh, does it bond other to a glucomolecule differently? Because exactly, Shawnees. Because now you have the hydroxyl in a different position, the bond inevitably will be different. Okay, because now that bond, the glycosidic linkage bond, is a bond that is created by an oxygen that traps the two sugars. So the position of the hydroxyl determines the bond. So what you end up getting is this difference. When you had the glycosidic linkage in the alpha glucose, like we learned before, this bond, the 1,4 bond, is going down, 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 down. So what happens is the polysaccharide has this bond that is going down, 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 down. Meanwhile, in the cellulose, so this is in starch, in the cellulose, what occurs is you'll have one going down, one going down, one going down, one going up. It's a zigzag. Okay? That's just to do this difference. Okay, so in starch, it's all going down, down, down. In cellulose, it's up, down, up, down, up, down. Now let's take a minute to think about this, okay? So this goes back to what, who was it that mentioned this? Wait, I'll tell you. I'm going to go back and remember. Oh, who was talking about the curly fries? I forget. Who mentioned the curly fries? I cannot find it. Remember, you asked about the curly fries? How come it looks like curly fries when you see the polysaccharides, the starch? Who was it? Anyways, okay, I was saying. So you have, I think Sunny did. Okay, so Sunny, you were saying curly fries. So I want you to think of this. You have the bond that goes down, 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 versus the cellulose where the bond is a zigzag. What do you think will happen to the overall molecule in the example when you have the bond is going down, down, down versus the zigzag? Exactly, Sunny. So it's going to be make it straight, right? If you're going up and down, up and down, up and down, the whole molecule will be like that. But if you have it going down, 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 it's going to force the molecule to make the curly fries, right? What do you think will be the difference of the material that you made with the curly fries? I'm going to call starch the curly fries now. With this versus the other guy. Okay, so I know. So what I will go back, I'll say it and show what I mean. Exactly, Sean. It's a good job. So what happens is when you have the starch is made of alpha glucose. Alpha glucose, because of the position of the hydroxyl down here, it makes all its bonds like this, going down, 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 down. This forces the overall polysaccharide to make this kind of curly shape that you see here, right? It's kind of it's flat. It's hard to see, but it's basically curling onto itself like curly fries. And while when you have the other example, cellulose, because it contains this beta glucose with the hydroxyl going up, you obtain bonds that are going up and down, up and down, up and down, like a zigzag, which makes the molecule really, really straight, really flat. And this allows for the molecule to behave differently. What happens if you have something like this versus something like this? What's going to be different? Oh, Theresia, we'll talk about straight and curly hair when we learn proteins, because hair is made of proteins, and we'll learn about that. Exactly. Not so much breaking apart, but it's going to be more compact, right? Because you have like this thing is going to be packed, while the other one is trapping all this air, right? So now think of this: the starch is potatoes, the cellulose is the bark of a tree. It makes sense, right? Because it's compacted, it's like dense. That thing is solid, while the other one has a lot of air. Starch has a lot of air in it because it's like all this space that you've created. Yeah? Does that make sense now? And Nicole lost her Wi-Fi for a minute. So, Nicole, are you lost or are you with us? Okay, I'll go back and reiterate, and that nobody gets hurt by this because it's helpful. So let's go back and see. So what we were doing is comparing structural polysaccharides that have the function of making a molecule protected. And in the case of cellulose, which is an example of a structural polysaccharide, cellulose is made up of a different kind of glucose. So glucose, when it makes a ring, sometimes it makes beta rings, which end up having the OH up instead of down like before. The beta rings, because the OH is up, when they make the long polysaccharide, instead of having all the bonds, glycosidic linkages going down, 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 they end up having a zigzag shape of the bonds, right? Because the OH going up makes this. So that zigzag shape of the bonds means that overall, when you have the usual starch bonding, this one, you will end up having this shape, a curl overall in the molecule. They will make like a loop. But in cellulose, where you have the zigzag, the cellulose is going to be very, very straight and compact. And this is why this is going to be such a dense mass. This is why when you compare bark of a tree versus potatoes, the bark of a tree is very strong and dense, and potatoes are soft and have a lot of air inside. Okay? And Shanice is saying, our beta rings only beta. It's the other way. Beta glucose rings are the bulk of cellulose. I think they can make other materials, but that's the one I know. Why is the so Sebastian is saying, why is the starch curly? Because if you go back and you see that this bond is always in the same direction, that forces the molecule to turn, right? Because it's always in the same direction, so it creates a tension, so it's going to force the molecule to make these loops. But if it's going zigzag, the tension created by one is limited, is resolved by the other one. So then it ends up being overall straight. And Olivia is saying, so the loop starches are loosened. Yeah, they have more air, Olivia. They have air in between, which are same direction. Which, what do you mean? The zigzags? And, yeah, exactly. In these, if you create tension in one that goes up, you have the opposite tension in the other one that goes down. You end up overall getting a molecule that looks straight, not curly. Now, there is another factor that makes structural polysaccharides also a very strong material, okay? And it's the fact that most creatures cannot break the bond that a beta glucose makes. Okay, so let me go back and show you. So beta glucose is, this bond cannot be broken by most living things. Okay, there's only very few creatures that can do it. So some microorganisms can digest this because we don't have the enzymes to break it. We can break starch, no problem. We cannot break cellulose, okay? And this is the reason, for instance, doctors would say, oh, it's good to consume a lot of fiber, because what you basically are doing is consuming a lot of food that you cannot digest. If you cannot digest a lot of foods, what are you going to be making lots of? Yeah, exactly. Poop. So when doctors want to say, okay, exactly, I don't say a person says, oh, I don't go to the washroom regularly. Well, if they consume a lot of starch, you are encouraging them going to the washroom regularly. It's helping them because you have a lot of waste. Exactly. And in connection to waste, I'm going to tell you something else that you'll like. So we don't have the microorganisms that break down this, so this becomes waste, but also some of the microorganisms that help us digest foods, help us digest some foods that sometimes create problems. So sometimes, for instance, so it's fiber, no, fiber, juice wrap. Fiber is good for you. You cannot digest it, but it helps your digestive system to flush in another way because you are basically getting rid of everything. I think it's good in good proportions. Yeah, yeah, it's healthy to consume salads. They have a lot of veggies, a lot of things, celery, plants, cereal, things that are not just flour. Yeah. Now, talking about waste, the other thing that is interesting is that when people produce farts, and everybody goes, "Oh, you're disgusting," it's not really you. Okay, what occurs is that when you consume things like beans or corn, you cannot digest that food. That food is digested really by the microorganisms you have in your colon. So those microorganisms are going to break down that food, and their byproduct of the breaking out of that food is methane. So if you want to have an excuse next time you do that, you just say, "It's not me; it's my microorganisms," and then you are exempted from it. But anyways, I digress. Let's go back. So we were saying, so the reason cellulose is very strong is because number one, it's very compacted, but also the other thing that occurs with this bond is that we cannot break them down. We require assistance. And even herbivores like cows require microorganisms to be able to break down plants. So even when you think of animals like termites cannot consume, even though they live on trees, they cannot digest it. They use fungi to digest it. So many herbivores cannot really digest plants. They need a microorganism to do it. Now this chitin is another structural polysaccharide. This is the polysaccharide that makes up the exoskeleton of insects. So when you think of a lobster, that outer shell is made of the polysaccharide chitin. So that's also a very strong exoskeleton and a very strong polysaccharide. This is the monosaccharide that makes the chitin. And sometimes they use it synthetic versions of chitin for sewing, for stitching, stitching. Sorry, stitching wounds. Okay, so I'll tell my mom that next time. Explain to her that you're learning a lot in biology. Okay, so structural chitin, da, da, da. Do we understand how herbivores need assistance to be able to digest this stuff? That's the end of carbohydrates. So I would love to hear any more questions, because if you don't have any more questions, I have a mini practice quiz for you guys. So any questions? Okay, I'll show you how to kindly repeat the part. Which part? What we're doing? Can you go over the shapes one more time? Just check it out. Yeah. So okay. So there is the two types of carbohydrates that we discussed. This is structural carbohydrate called cellulose. Cellulose is made up of a bond that is created by the beta glucose, and this bond ends up not just going down, 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 but it goes up and down, up and down. So their glycosidic linkage behaves differently. And because this glycosidic linkage has a zigzag shape, cellulose ends up being, sorry, let me mute this because it's such a pain in the cellulose ends up being straight, and that makes it very compacted. Okay, and the other thing that matters is that the bond that beta glucose makes, we cannot break. Most herbivores cannot break, and only some microorganisms can break. Cool? With the herbivore? Herbivore? Can you go over there? Yeah. So the herbivores require assistance of microorganisms that live in their chambers. This is why cows have like 3 or 4 chamber stomachs, because they have to kind of grow those microorganisms in there and allow. And this is why ruminants ruminate. They have to get the food, basically stir it up so all the microorganisms have access to it. Yeah, they're trying to grow, for instance, cows when they're babies, when they're born, sterile without microorganisms, and they cannot eat grass at all. So they cannot eat grass. They have to have that help. But so much methane. Yeah, Olivia, well done. Exactly. Exactly. It's the microorganisms that do that. So, and let's face it, that's a severe contribution for climate change. Yeah, it's a big problem. That's why climate change, what people say, you should go vegan. Do you even grow a sterile cow? For experimental purposes, they've tried that. They basically take the baby, comes out of the mom, and they put it in a bubble of plastic with no microorganisms to do this experiment, which I think is like cruel but interesting. Okay, I think we're good for our D2L quiz that I made. It's only 5 questions, and it's just to see how we're doing, and also mostly because I wanted to try D2L. So follow me, I'll show you what you do. So you go here, you go to this link with d2l.com, so very easy, and then you're going to get logging help, and then your organization is YRDSB, and then you go there, and it's going to have student login. For me, it's already logged in, but you will student login and you find our course, which is this SIC 09 Biology German, because our section is 09 and it's under science. I try to get rid of SIC, but it doesn't do it. Okay, so tell me if you are succeeding in doing that. Please tell me if you are having trouble with that. Got it? Tell me. Got it, got it. Nice, nice, nice. Anybody with trouble? Okay, beauty. Sebastian has trouble. No, why? Did you go? Go here, d2l.com, logging help, write YRDSB, and then student login, and you should be able, no, don't go there. That's not our class. This is our class. Keisha is fine. Sebastian, try again. Let me know if you have trouble. Shanice is fine. Simabri is fine. Tammy, Trisha, when I tried to log in, it said your username was asking, but Trisha, yeah, because when I tried to enter your name and your student number and your last name, none of it worked. I don't know why. I haven't had that in all the other classes. Like, you know, I have almost 100 kids, and you're the only one that had that. I don't know why. So I forgot to do that. I have to ask somebody why you are not in there. So, sweetheart, I am so sorry. I will figure it out. What do I press for school again? YRDSB, York Region District School Board, YRDSB. Thank you, Simaver. Oh, Diana, that's very kind of you. Trisha, won't hurt if you mail guidance and said, I don't seem to be able to access D12. Yeah, try that. And I apologize that you cannot do this. But I'll show you afterwards. We'll take it up and you'll have it though. Grace, once we are, okay, let me show you what you're doing then. So once you're here, you're going to go to content. And what you would see in content is not going to be exactly like me because I can see, because I made the course, but you will see course of review biological molecules, and in here you will see carbohydrates, and then in here you'll see carbohydrates, and this is a quiz. I should have called it carbohydrates quiz, but why did I do that? I don't know, because I had to make it like 5 times because D12 sucks. I honestly have made this quiz so many times. My period 1 class tried it twice. And you cannot save it or edit it. You have to remake it. It's a pain in the neck. Okay, go ahead, do it. And hopefully this time it will work. No, because you cannot edit it. So you have to remake it from start. But if it works with you guys, I have it. Okay, so go for it, guys. And we'll take it up in a minute. So Sebastian, what you should see is called carbohydrates. It doesn't say quiz or anything. It's just called carbohydrate and has a question mark. I don't know if you can do that. I think if you go to content and you see the word carbohydrates with a question mark, that's the one. The username for D2L, I think so. I think it's your student number. The number is not yours. I don't know what it does that, Olivia, because it puts a student username and sometimes the number is not your student number. Don't worry, I think that's still you. Don't worry about that. Olivia, go ahead and do it anyways. That's you. Yeah, I don't know why it does that. I just see the page of carbohydrates. No, it has to say carbohydrates. Okay, Sebastian, look at me. I'm going to show you. You have carbohydrates. You click on it. You open content. You won't see this because this is locked for you guys. This is your quiz. You open that, go to the quiz. Got it? So yeah, go start. There's only 5 questions. Oh, clever boy, Sonny. I get it. It's your OEN number, Olivia. I didn't know this. I have no clue. A very, very important number. Okay, so, okay, guys, just do the quiz. It's 5 questions. We're going to lose time, and I still have other things I wanted to do. I will take it up after. We'll do all of them. Okay, so there's only five questions, so you don't have that much time. I have like 10 minutes, but I'll give you a few more minutes. When you are still working on it, let me know because I will take it up. It's supposed to show you your score, but don't worry because this is honestly just for us to take up and check. Yeah, I don't know. I still have to figure out how this thing works, truly. Okay, let me know if you still need a bit of time. I'll give you a few more. Yeah, that's right, Olivia. At least it works, but it is. I always prefer Google Forms. Okay, guys. Um, I want to take it up before our period finishes, so let me know who's still working on this. I'll have a look. Hopefully good. Okay, great, Sebastian. So try to wrap it up and then we'll. Oh, geez, I'm sorry. Um, and I'll go back and we'll take it up. Oof, what happened? No, miss, like you. No, miss, I like you, but it I, I like with you, but summer school was okay. Oh, I'm sorry. I didn't like it. Oof, what happened? What do you mean, what happened, Zoe? Did you finish your quiz? I haven't checked. I just submitted. Okay, okay. No, no, we're waiting for a little bit of time. Oh, what happened? Summer school is tough though because so quickly. Oh, jeez. I don't know. It all sucks. Don't worry, Sebastian. We'll take it up. Okay. Yeah, it's super fast, right? Because you're trying to cover a lot of territory in a very short period of time. So it's a bit struggling to do it. But sometimes people like to kind of get ahead and take some courses. Truly, I mean, this year, in a way, it was a good year to do summer school because of the quarantine. But on the other hand, the summer was when restrictions were a bit lighter. Some kids prefer, like my daughter, I told her, why didn't you get a head start? And she was like, I cannot do this anymore. And she needed a break from the whole thing. She found it really hard. So, yeah, it depends. She's in grade 12 like you guys. So okay, how we doing? Should we take it up? Let me have a look at our schedule. How people are. So I think that if we don't take it up soon, we won't have a chance. It's horrible, haha. Oh wow. Yeah, sorry. Yeah, it's tough. Okay. Are we good to take up this quiz, guys? Cool. Okay, let's do it. I'll tell you a story that will top up your thing of having so many assignments. In the TDSB, not in our board, in the TDSB, yeah, I will, Thrich. I'm just going to tell you the story quickly. In the TDSB, they've done something called quadmesters because of COVID. So kids have to take like a course in half a semester. So basically, they have like a test every week. It is so stressful. Yeah. Okay, I'm going to share my screen. Yeah, it's so stressful. A friend of my daughter's is in a TDSB school. She's like, find it so difficult. Okay, quiz. No, that's not my class. That's your class. Let's go to the quiz. So these are our questions. Which carbohydrate is a storage carbohydrate? So these questions are tricky. I made these questions. And so the option you have to take is which is a storage carbohydrate? Glucose, ketone, cellulose, or glycogen? And glucose is not a storage molecule, right? Glucose is just for energy. Ketone is a structural carbohydrate. Cellulose is a structural carbohydrate. So glycogen is the next answer, the right answer.
But we can't see.
Oh, wait, the sharing didn't work. Thank you for telling me. Let me see why the sharing is not working. Let me see if now it works, okay? Can you see my screen now?
Yeah, but we can't see the answers.
Oh, no, I just will show you. Well, which carbohydrate is a storage carbohydrate? So it won't show you, this is to do with D to L. I'll show you the answer. So the answer should be, the only one that is storage is glycogen. Okay? Because ketone is structural, cellulose is structural, glucose is just a molecule that gives you energy, but not stores energy. Okay? So if you had that, that's your right answer. Then the next question says, what is the general name of monomers of carbohydrates? And the answer should be monosaccharide. And I'll see why it doesn't show you the answers, because that's a glitch of the program. Then, what is the name of the linkage between monomers and carbohydrates? And that's glycosidic linkage. What functional groups are found in monosaccharides? We've done this question before. And the answer should be hydroxyl and carbonyl. The last question of all of them is, which type of carbohydrates is most likely to be used for structural purposes? And this is a tricky question because you will choose cellulose probably, but that's not a kind of carbohydrate, that's a specific carbohydrate. So the answer should be polysaccharide. Okay? So, okay. So now, do you have any questions about this quiz? Any questions about the options? Any question about what you had to choose? Any part that was confusing? The last question is a bit tricky. It's saying which kind of carbohydrate is likely to be a storage carbohydrate. So if you are impulsive, you will choose cellulose. Cellulose is not a kind of carbohydrate. It's a specific name of a carbohydrate. Okay? So the kind of carbohydrate is polysaccharide. So that question is a bit tricky. That's and what's the answer for the first question? So I don't know which is your first question because you will not get in the same order as everybody else. So which one? Natasha, just the one that is saying which is storage, glycogen. Natasha, if that's the one, glycogen. Yeah. Okay, I don't want us to finish the lesson without showing you what's going to be your practice for tomorrow. So tomorrow we're going to do a whole period for inquiry where you plan your lab and I'll give you a mini practice quiz for inquiry. So I want you to try to do some of these. We'll take them up and discuss them also in class tomorrow, but I posted in the Google Class this. So let me show you. In the Google Class, I posted this. I went to variables practice. And this handout, which is not a Google document, it's a paper, it has examples of experiments that you have to see if you can label which is independent, which is dependent, which is control. And I think that you guys could use some practice for that. So, in fact, I would say we don't have enough time to do practice now because it took us a bit longer than I expected to do the D12. But try to do that one at home. Maybe not all of them, but a few, and then we'll discuss them in class tomorrow at the beginning. Yeah? And I'll see if I can release the scores for D12 so you can see them. Let's see if I can do that. What's the difference between what I mean is this? Cellulose is a specific carbohydrate. It's not a kind of carbohydrate. A kind of carbohydrate is monosaccharide and polysaccharide. Cellulose is a polysaccharide. But there's many storage polysaccharides. They all fit into polysaccharide now. Okay, so when you say what type of carbohydrate are we talking about? We're talking about polysaccharides. There's many storage polysaccharides keeping cellulose. That's the difference. Could you pull up the last question again? Could you give another example? Another example of a polysaccharide that is storage? Olivia? Names of kind. For instance, you can say which kind of carbohydrate is likely to be an energy source, an immediate source of energy for consumption. The answer should be glucose. It should be a monosaccharide or a disaccharide. Glucose is an example of a monosaccharide, but any monosaccharide could be a potential source of immediate energy. Yeah, it's more an English language thing than it is actually about chemistry or biology. So, Sunny, do you want me to go back to the last question and show you quickly before we leave? Yeah, and so let me show you, where were we? This. So this is the last question, and it's saying, which type of carbohydrate is most likely? See, it's a type of carbohydrate. It's not the specific carbohydrate. So polysaccharide is a better answer than cellulose because it's more likely to be used for structural purposes. You cannot say that for cellulose. Cellulose is a structural carbohydrate. Polysaccharides are more likely to be used for structural purposes as opposed to monosaccharides. It's just a wording thing. Couldn't polysaccharides be used as storage like starches? Yes, but it doesn't say exclusively. It says it can be used for protection as opposed to being a monosaccharide, but it doesn't mean only. Can also be storage. Does that make sense? It's wording. Cellulose is only used for structural purposes, right? Yeah, cellulose is only a structural carbohydrate, yeah. And it doesn't mean that some things won't eat it, but it's a structural. Wouldn't it be a better answer? Cellulose as a choice? The question asks about structural purposes. What do you mean by structural purposes? It means it's a structural carbohydrate as opposed to a storage carbohydrate. So the whole confusion is that cellulose is not a type of carbohydrate. It's a specific name. It's like saying, let me see if I can find an example. Which type of TV show or YouTube show doesn't require acting skills? Your answer should be a reality TV show. It shouldn't be the Kardashians, because the Kardashians is one of the reality TV shows. Does that make sense? That was for the last one. Okay, guys. Now we have to wrap it up because it's 2:54. No problem. I was trying to connect with the kind of shows you guys may watch. I personally never watched the Kardashians, but as my daughter, she knows all these women and all their crap. Okay. Okay, guys. So have a good afternoon. Try to do that handout. Go through your notes. Lots of information. No clue, sorry. You're asking the wrong person. But yeah. So yeah, have a nice afternoon, guys.
Miss, I have a question though.
Uh-huh.
It's about that curly, the curly fry thing we were talking about again. Can you go back to that slide real quick?
Yeah.
Um, you know when it says like you said that basically starch is basically it's curly, right?
Uh-huh.
Cause you know how you say because it goes in one direction, like you know how like it just goes in one direction, right?
So, uh-huh.
Yeah. What do you mean?
So
Like, what goes in one direction? Like the OH?
it means that all the bonds are like this. They're going down.
Oh, the
So
bonds, right?
yeah, all the bonds are going down. See, all the bonds are going down, 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 down. So that forces the molecule to curl.
Oh, but which bonds specifically? Do you just say bonds or just like it just
The linkage bond between all the sugars that make up starch is always this kind. And so it's always this way. And because it's always going down, it makes the molecule curl and curl and curl.
Oh, so basically it's the linkage bond that makes sugar that it's the bond that goes like that's always in the same direction. It's basically the linkage bond that makes sugar, right?
Correct.
Oh, okay.
In that molecule, in the starch molecule, is always that way, but in the cellulose molecule, is one is down, one is up, one is down, one is up.
Oh, okay.
So that curl.
And you know how it says what? So does it make the molecule curl or just the bond curls?
The overall, the bond itself is rigid; it's not going to bend.
Mm-hmm.
But the overall pressure that it creates on the long chain makes the overall chain make loops. So each of them,
yeah,
creates tension, right? Because it's going down, down, down. So then the molecule will eventually have no choice but to loop.
The curly.
Because it's kind of pushing the molecule down.
Yeah. So it's basically like the molecule gets curled because the linkage bond of the sugar goes down. Like it's in one direction.
The whole macromolecule gets curled. The bond is not going to change direction. The bond is fixed.
Yeah. And then it's just basically the opposite for the Celestis one because basically the linkage bond that makes sugar just goes up and down, so that's why the molecule is just straight.
Exactly.
Oh, okay. And for the cow one, the cow like the herbivore thing, you're just saying that basically the herbivores require more assistance of microorganisms in there, right?
Because the bond that goes up, we cannot digest it, and most animals cannot digest it, and only some microorganisms can break it down. Oh. So that they cannot help it. They need assistance to break that bond.
Oh, so basically they need more assistance microorganisms in order to break that
Because
bond.
they don't have the enzymes to break those bonds. They don't have it. So they need like a foreign entity to do it.
Oh, but it's to break the linkage, like that sugar thing we were just talking about, right? That bond.
The one that goes up, yeah.
Oh, so it's always for them. It's always going up.
Well, one down, one up, but the one that goes up, they cannot break.
Oh,
Unless they
okay.
had.
So they basically have the one up and one goes down, but the one that goes down they cannot break. So that's why they need more assistance in their microorganisms to break it down. Boom. Okay, I get it now. Yeah.
No problem.
That's it, miss.
No worries. So I'll see you tomorrow.
Yeah, see you tomorrow. See you, miss.
See you later.
Check